Hi all, this is the fourth mini lecture of chapter 13. Hopefully these are helpful and not driving you all crazy. The learning outcomes for 13.4 are here. This is mainly covering the T cells, so we'll get into that, the T cell response. So we'll start out with, now we've done stages 1 and stages 2. We're going to cover stages 3 and 4 in this mini lecture, which is the T cell response. Remember, the T cell response is a cell-mediated immunity, and that the T cells are really a complex part of the immune system, and they have a huge number of types of T cells that play a role in this. So when we look at the T cells, we know that we have to have an antigen presenting complex that allows them, or an antigen presenting cell that presents the antigen to the T cells. And depending on what they're being presented by, if it's an MHC1 presentation, then that's going to activate CD8 cells or cytotoxic T cells. An MHC2 will activate that CD4 receptor on T cells, which are those helper T cells generally. The T cells then can produce cytokines that have a large amount of biological effects on other parts of the immune system. And at the end of T cell stimulation, what you should see is a mobilization of many other types of T cells, that stimulation of B cells, and then of phagocytes, so cells that can go ahead and continue to phagocytize whatever the foreign particle or cell is. So some of the interesting different subsets of T cells, and I'm not showing all of them, but these are kind of the really important ones that we might see. We have a number of different T helper cells. And in this case, all the T helper cells and the T regulatory cells use a CD4 receptor, and it's just that cytotoxic T cell that uses a CD8 receptor. So for these helper cells, it may be that they're going to secrete cytokines, or look at different types of allergy occurring after several hours or days of contact. It may be ones that can help with that B cell proliferation and secreting interleukin 4, 5, and 13. We also have the T helper cell that can promote inflammation by secreting interleukin 17. And then those T regulatory cells are really important in making sure that we don't get too big of a response, so kind of regulating it. And then last, those cytotoxic T cells, which can do cell destruction or destruction of foreign cells. So our T helper cells, I already talked about that CD4 is their primary receptor, and they're also involved in activating macrophages and then stimulating that proliferation of T cells and B cells. With cytotoxic T cells, these are the ones that are going to destroy target cells, and these can be virally infected cells. So viruses might infect a cell, and then their peptides might be displayed on the cell surface. The cytotoxic T cell will recognize that and destroy them. Cytotoxic T cells can destroy cancer cells, so they recognize abnormalities on these cells and recognize that they need to be killed. And the last, they can recognize cells that come from another animal or human. And this would be important in something like a graft rejection. They're going to recognize something as being from outside the body. Remember that cytotoxic T cells are the only ones that have the CD8 receptors instead of a CD4 receptor. There are a few cells that are a little bit different. They kind of straddle the line between the non-specific immune response and the specific immune response. We have some cells called gamma delta T cells, and these are different in the sense that they do not necessarily have to have an antigen presenting cell to be activated. So they have receptors that actually allow them to work a little bit like those white blood cells did on recognizing PAMPs, which means that they're not specific like we see with normal T cells. However, they can produce memory cells, which is why they kind of straddle that line. They don't have the specificity, but they do have the memory portion. This does allow them to respond quickly in the sense that they can recognize any foreign or certain kinds of foreign molecules. They're very responsive to phospholipids and also to tumor cells. And as I said, these bridge that divide between nonspecific and specific, since they have memory but no specificity. We have a few other cells that can kill particular infected cells, much like a cytotoxic T cell. We have what are called the natural killer cells, and these are not a true part of that specific immunity because they don't have specificity, but they can attack cancer cells and virus-infected cells, and they work very similar to a cytotoxic T cell without the specificity. And they also don't have memory cells, but they are affected by some of the cytokines that are being produced, which would put them as a little bit part of straddling that specific immunity. The last type of cell that's a little bit straddling the nonspecific and specific immunity is natural killer T cells, or NKT cells. These are like the killer cells that we just talked about, but also like T cells. They actually have T cell receptors for antigens, and they can release cytokines, which is a hallmark of those T cells. They can also kill cells, so they're very quick to do that. And they do not have memory cells, though, so that's again why they are between nonspecific and specific immunity. So when we're looking at that T cell response, this is a great table in your book, and this is one that if you really want to put things in kind of an organized fashion, I recommend you look at the T cell activation table. So at the top we have that antigen enters the antigen presenting cell, and it activates either the CD4, which is in the blue up here, or the CD8, which is in the white down at the bottom. And what will happen with a CD4 cell is that it can become a number of different things, and usually it will activate and produce many different types. So remember, we always say it has that memory CD4 T cell that is like the T cell that recognized the antigen. It will then produce different types of helper T cells, ones that might activate the B cell line, helper cells that will help with tumor inhibition here and stimulating macrophages, also T helper cells that can activate our inflammation response, and then lastly those T regulator cells that can actually tamp down and be careful about the reaction that we have and make sure it's appropriate to what the stimulation was. So this would all be those CD4 cells, and this would be with an antigen presenting cell. Now if we look at the CD8 cells, and remember these are the cytotoxic T cells, with that antigen presenting cell on an MHC1 versus the MHC2, we're going to get some helper T cells. But then what we're going to look at really getting is those memory CD8 T cells and then an activated CD8 cell. And these are what are the ones that are capable of going out and killing or breaking down, destroying some sort of a host cell that has been infected. And we see here that that CD8 molecule will once again recognize an MHC1 presenting antigen, and it will break down the cell that has that infection in it. So those are really the two different T cell activations that you need to know. And last up is that checking in, talking about what would activate a T cell, and also being able to identify which cells have CD4 receptors or CD8. And then a little bit more on T cells or cell types that fall between the T cells, or sorry, the specific and non-specific immunity. Hope that helps.